This video is going to cover the physiology of blood. The learning objectives that we'll cover, we'll start with a brief introduction to the components of blood, a little bit about erythrocytes and leukocytes, and finally blood clotting and blood typing. So the major functions of blood, obviously blood helps circulate things around the body, so transportation would be one of the major functions of blood. Things that are transported around your body in the blood are things like the gases like oxygen and carbon dioxide, but also nutrients like fats and hormones and glucose and all types of things. We also circulate cells around and proteins, ions that we've learned about, sodium, chloride, calcium, all the things that are outside our cells are circulated around in the blood. The other cool thing about blood is that blood clots up when our blood cells are damaged or when the blood stops or slows. So that way when we have a leak in a blood vessel or damage internally or externally, that blood will clot and stop the blood loss from our blood vessels. And it's not magic. We'll cover the steps. I wanted to remind us that the pH of our body's fluid is very important, and since the extracellular fluid basically mixes with our blood and our blood plasma, all the sodium, the chloride, the hydrogen ions floating around will affect that. So when we have too many hydrogen ions floating around in our extracellular space, that will tend to cause our pH to go down. So a low pH, we remember, is acidic. So again, anything that's happening in your extracellular fluid happens as well in your blood. We have lots of defense cells circulating around in our blood called leukocytes. We also have defense proteins called antibodies, which we'll learn about. The blood, we know, is important in temperature regulation because we change our blood flow to our skin depending on whether we're hot or cold. All right, what is in blood? What are the components of blood? So let's start with plasma. Plasma, as you'd expect, is mostly water, and then we have to add some special things like sodium and chloride, calcium, maybe a little bit of potassium, and other ions to our water. So basically, plasma, the most basic component of plasma, is saline or sodium chloride, and if you put in 0.9%, that's basically the exact mix of water and sodium and chloride in your body, in your extracellular fluid, and in your plasma. So again, don't forget, in our extracellular fluid we have more sodium and more chloride than inside our cells. We also have dissolved gases in our plasma like oxygen and nitrogen and carbon dioxide. Those are dissolved gases we find in the plasma and in the blood as well. Another key ingredient in plasma and in blood are these plasma proteins. So the most famous plasma protein is albumin. There's some other clotting proteins which we'll learn about. Albumin's really interesting in that it attracts water. That helps keep water inside our leaky blood vessels. And the liver makes many, probably not all, but many of our plasma proteins like albumin and the clotting proteins are made in the liver. We have lots of nutrients like glucose, fatty acids, cholesterols floating around in our blood, and again the liver helps make that cholesterol and even some of the glucose that we find in our blood. We also have hormones in our blood dissolved in the plasma or bound to plasma proteins, and we've learned some hormones already this year. Okay, we also have cells in our blood. And so besides plasma, now we have cells suspended in our blood. And the erythrocytes are the most common blood cells that we'll find. Erythrocytes' jobs to carry oxygen with a specialized protein carrier called hemoglobin. Hemoglobin has a little iron molecule in there, and that helps attract the oxygen. So that's the job of our erythrocytes, is to carry oxygen. And when erythrocytes and the hemoglobin has less oxygen, those blood cells look darker, and so blood is either sort of bright red or more maroon and dark red. Okay, in addition to erythrocytes, we have our leukocytes, or white blood cells. These are our defense cells to defend us against things like bacteria and virus-infected cells, cancer cells, anything that's bad that can invade us. Our defense cells, our leukocytes, can protect us. The white blood cells are in far less concentration than the erythrocytes. So the erythrocytes would give you your blood its red color and the hemoglobin. Okay, the types of leukocytes we have neutrophils, we have monocytes that once they're in the tissue we call monocytes macrophages. We have lymphocytes, and lymphocytes come in varieties, B cells and T cells. We have eosinophils. Eosinophils have this characteristic look when they're stained. They're really orange, so we call those eosinophils. And then we have these really deep purple stained cells called basophils. So those are the leukocytes. And we tend to have more neutrophils and lymphocytes and then less of the eosinophils and basophils. So those are more rare. And we'll talk more about the different functions of each of the leukocytes in another learning objective. But you have defense cells, some that attack other cells, others that make chemicals. Some of them may be great at creating immunity to infections and things like that. So we'll go through these in more detail in a little while. All right, neutrophils and monocytes are really good at attacking stuff and defending you. Okay, so if you've ever heard of B cells or T cells, those are types of lymphocytes. I just wanted to point out that B lymphocytes are famous for making defense proteins called antibodies, which we'll talk more about. Another type of, not really a cell, but a piece of a cell we'll find in our blood are called platelets. Platelets are for blood clotting, and if you're wondering where platelets are made, they're made the same place that all of our other blood cells are made, and that's in our bone marrow. So you have this cell called a megakaryocyte. Megakaryocytes are found in your bone marrow, and they basically bleb off little pieces or chunks of cells, chunks of themselves, and we call those little bits of cells platelets. So a platelet's not really a cell itself, although some textbooks call them thrombocytes. We're going to call them platelets to remind us that they're little bits of cells that can clump together and cause clots. All right, these are what your blood cells look like when you stain them with hematoxylin and eosin. Let's talk a little bit about the production and function of erythrocytes. So again, big picture, quick and easy thing to remember is that the function of erythrocytes is to carry oxygen bound up to this carrier protein called hemoglobin. And red blood cells have a traditional disc shape that's important for their function. Where are your red blood cells made? Same place all your blood cells are made, in your bone marrow by hematopoietic stem cells. Again, if we stain our blood, we can see those cells. We can see the white blood cells versus the red blood cells, many more red blood cells. And they lack a nucleus, so they don't have that stained purple nucleus like the white blood cells. Again, hematopoietic stem cells in our bone marrow give rise to all our different cells, whether they're leukocytes or erythrocytes. So hematopoietic stem cells can divide and go down a certain lineage to create our new little red blood cells. Red blood cells do not have a nucleus, and they do not have mitochondria once they're mature, and I think that's so they can pack themselves with tons and tons of hemoglobin protein to carry lots and lots of oxygen. So basically more room to carry oxygen. If you're wondering how these cells can make ATP to survive, well, they basically use glycolysis, which you know doesn't require mitochondria. So red blood cells use glycolysis to make their ATP from glucose. And they don't have a nucleus, so they don't have an instruction book, and they're probably not making lots and lots of new proteins once they're mature. Red blood cells have a pretty short lifespan, about 3 months or 90 days. Once they've been around a while and start to get damaged or misshapen, they're destroyed and recycled, usually by the spleen. So we make our new red blood cells in our bone marrow, and then we get rid of them in our spleen and maybe a little bit in our liver. So remember, we're making all these red blood cells in our bone marrow. We call that erythropoiesis or hematopoiesis. And you might wonder, how many blood cells does my bone marrow need to make in terms of red blood cells? Well, we have a hormone which can tell those hematopoietic stem cells how much they should be making new red blood cells. And that hormone is called erythropoietin, or EPO. So the kidneys act as oxygen sensors and then produce erythropoietin, which targets the bone marrow. So anytime your oxygen levels in your body get low, your kidneys will sense that, and those little cells will start making more erythropoietin, which will then tell your hematopoietic stem cells to make more red blood cells packed with hemoglobin, which will carry more oxygen around your body, and eventually that oxygen will reach your kidneys and homeostasis will be reset. So remember that the kidneys are in charge of making the erythropoietin. So what's so special about hemoglobin? Well, obviously it can carry oxygen and it's able to bind it and release it, which is really cool. So how does hemoglobin carry oxygen? Well, the hemoglobin proteins have these heme molecules, which you can see is kind of complex. We're just going to remember that the heme molecule has iron, and iron can reversibly bind oxygen. So each hemoglobin can bind 4 oxygen molecules. The heme part is the part with the iron in the hemoglobin. So this is the life cycle of an erythrocyte. Again, erythropoietin stimulates your hematopoietic stem cells. You make a bunch of new erythrocytes every 90 days. They're sort of tired out, misshapen. The spleen has these macrophages which know to eat up the damaged ones, and we sort of recycle them. Again, the spleen is the primary place. So we can break down the hemoglobin as we get rid of those red blood cells. The globin, basically we can recycle the iron and the amino acids. What about the heme? The heme has pretty complex metabolism, which includes the liver. The liver will actually process that heme into things like bilirubin and urobilin. Urobilin you can pee out from your kidneys. The bilirubin you can get rid of through bile in your poop, and so that's how you can sort of process the bilirubin and get rid of those heme molecules. If the bilirubin builds up, that's called jaundice, and we can talk more about that in class. So big picture again, the spleen is going to remove our red blood cells. The liver will help recycle that heme. The shape of our red blood cells turns out to be really important. They cannot be round like a sphere or basketball. They have to be disc-shaped to fit in our blood vessels and to carry the most amount of oxygen. So anytime our red blood cells are misshapen, that can cause destruction and damage and break apart those little cells, which can lead to destruction of our blood cells, which can cause anemia. And we've learned about sickle cell anemia, where you have a mutation that causes your hemoglobin to be misshapen, which causes it to clump together inside the cell, which causes them to sickle shape. So that's a hemoglobin problem and a hemoglobin shape problem, which changes your red blood cells. It all tracks back to your DNA, and there's a single mutation in the DNA which causes the hemoglobin protein to change shape, which then changes the shape of the cells, again causes them to sickle shape. Sickle cells can then clump and cause lysis, or breaking apart of those cells. So that's one way you can get anemia. Another way you could get anemia is simply by having a lot of blood loss. Destruction of your red blood cells, or a problem producing your red blood cells. For example, if you have cancer or kidney problems or some hormonal deficiencies or dietary deficiencies. Okay, let's quickly review a little bit about our white blood cells. Those are our defense cells, and again we said we had the certain varieties. We have neutrophils, monocytes, lymphocytes, eosinophils, and basophils. So these are our different types of leukocytes. Neutrophils are probably one of our quickest and most rapid attackers. So they can attack bacteria, infected cells, sick or damaged cells, and cause lots of death and destruction. So much so that sometimes they'll kill some of your healthy cells. So neutrophils are very powerful. Monocytes are kind of like a neutrophil in that they like to attack about all the same stuff. They probably last a little longer once they get into the tissue and they're out attacking. Neutrophils tend to die off pretty quick, maybe within a few hours, whereas monocytes might continue to attack and defend for a few days. Both of them are considered phagocytic cells, so they eat stuff up, which we call phagocytosis. So they can actually ingest a bacteria cell and actually then destroy it. So we call that phagocytosis. And again, both of these are good attackers at killing things. Neutrophils actually make some killing chemicals which can help destroy cells and invaders. Lymphocytes, we'll learn more about those when we learn about immunity in a different lecture, but I just wanted you to think about them as some of your strongest defenses, specific defenses with immunity and memory. And we have different types of lymphocytes, T cells and B cells. The B cells make antibodies, and they create your strongest defense, your lymphocytes. Eosinophils are sort of interesting in that they think they've evolved to attack and defend us against parasites. So parasites would be things we could get from our food or water, but sometimes we're not exposed to lots of parasites, and then those eosinophils seem like they don't know what to do, and they start causing things like asthma and allergies. Basophils, they're the rarest of the leukocytes, but when they get activated, they make chemicals like histamine. They also make heparin. Histamine causes a lot of those things you get when you get sort of an allergic reaction. A similar cell to a basophil, but different, is a mast cell. And it makes histamine. Histamine gives you all that swelling and itching and stuff like that you get during your allergies. So they're trying to help you, but sometimes those chemicals sort of annoy you. If you want to remember the most common to least common leukocytes, if you did a blood cell count, is never let monkeys eat bananas. So neutrophil, lymphocyte, monocyte, eosinophil, basophil. So if you were to do a white blood cell count, you'd expect mostly neutrophils, lots of lymphocytes, and then less monocytes and rarely eosinophils and basophils. For the next learning objective, we're going to cover the major mechanisms of hemostasis, or blood clotting. So it's actually an interesting property of blood that whenever our blood vessels get damaged or injured and they start leaking blood, the blood actually helps clot up and stop the bleeding itself. So let's say you have a damaged blood vessel, and so you have damage to a blood vessel, and the bleeding will either be internally or in this case externally in our fingertip. That damaged blood vessel will keep bleeding, but luckily our blood is going to help stop that blood loss, called hemostasis. So the first step in stopping blood loss will be vasospasm. It's a tendency for an injured blood vessel to vasoconstrict, and that probably helps slow down blood flow, which will help promote clotting. A next step, and a very important step, is the activation of platelets. So platelets are normally floating around inactive, but whenever we have damage, they become activated, which causes them to clump together and to become sticky. And as they clump together, they form a platelet plug. So that's an important step in hemostasis. And then the next step, which might be the most important step, is going to be the activation of clotting proteins. Clotting proteins are normally inactive in our bloodstream and in our plasma, and only when there's tissue damage or blood vessel damage do we activate those proteins, those clotting proteins, in a complex cascade to create a fibrous clot. And so that's an important step in hemostasis, the blood clot formation. Again, it seals up that blood vessel and stops the blood loss. So again, the first step to help slow down the blood flow will be vasospasm or vasoconstriction, which will help in the hemostasis process. A next and very important step is the activation of platelets. So again, platelets are normally inactive in our bloodstream, and so then when we have damage to our blood vessel wall, that endothelium gets damaged, the collagen and the basement membrane underneath are exposed to the blood, and this causes activation of the platelets. The platelets then begin to clump and cling on to the damaged portion, and we call that the platelet plug. The next and final part of hemostasis is the clotting cascade, where we activate inactive proteins that float around in our bloodstream. We call it the clotting cascade because it's several steps of very complex reactions. We're going to focus on thrombin and fibrin. So again, blood vessel damage exposes the collagen underneath the endothelium. Platelets begin to plug and platelets adhere. We're going to talk about activating some of these clotting proteins. So the most famous ones, again inactive, are prothrombin and fibrin. Well, when we have this damage, we actually activate the first or most important step for us is going to be creation of the active enzyme thrombin. So thrombin is an enzyme protein which when activated can actually interact with fibrinogen. And when it interacts with fibrinogen, it cleaves off a little piece, creating fibrin. Fibrin is actually a protein which can clump on to each other or polymerize, and these little fibrin molecules then create this protein meshwork or protein clot. So the fibrin clot is created by thrombin, and again, this was all activated by the damage. So then we get a stable clot with platelet and fibrin protein. So damage activated this complex protein cascade, which activated thrombin, which helped create fibrin. And so that's the clotting cascade. And there's several steps to it. We're going to focus on thrombin and fibrinogen. So whenever you have damage to your blood vessels and endothelial cells, it creates this very complex reaction that activates the enzyme thrombin. Thrombin cleaves off a little piece of fibrinogen, creating fibrin. Fibrin likes to polymerize into a meshwork, or think of like a net of protein, and that's the actual stabilized clot. The liver actually creates fibrinogen and prothrombin, which circulate around in an inactive form until you have some type of damage to initiate the clotting cascade. What if you want to block clotting? An easy way to prevent clotting or to reduce clotting is to take a daily aspirin. A daily aspirin causes permanent inactivation of platelets for their 7 to 10 day lifespan. And so aspirin actually helps prevent or reduce clotting by reducing the number of activated platelets. If you want to use a more powerful anti-clotting drug, you might take something like Coumadin, warfarin, or the newer Xarelto. Those block the clotting proteins or the clotting cascade. When you do that, you block the activation of thrombin and the creation of fibrin. And so again, that's a powerful way to prevent blood clotting or reduce blood clotting. The downside of taking these drugs is that sometimes you might bleed internally or externally and bruise. So basically, when you're taking anti-clotting drugs, you don't want to bleed to death, and so you want to take as little as possible to prevent maybe your stroke or heart attack, but not have the side effect of bleeding too much. If you've heard of hemophilia, these are people that often can't form blood clots or have reduced clotting. This is often due to DNA mutations which cause the clotting proteins to not form correctly or not behave correctly, which reduces fibrin formation and the ability to form blood clots. Again, the downside of this would be internal bleeding and damage. The final learning objective, we're going to cover blood type and blood transfusions. So what does it mean to know your blood type? Well, blood type is based on markers that our little erythrocytes make. And so markers, if you want to think of them, are little identification molecules that can be carbohydrates or proteins. All the cells in our body make little identification markers that our defense cells can check and see if your cell is healthy and belongs there, or to detect cancer. We call these markers antigens. Antigen is really just a marker that our defense system can detect. So normally we want to ignore our own markers. So when it comes to erythrocytes and blood typing, we're concerned with 3 different markers that people can make: the A marker, the B marker, and the RH marker. And sometimes we'll call those markers antigens. So there are 3 possible antigens or markers that we're going to worry about with blood type: the A, the B, and the RH. A and B refer to carbohydrate markers made by enzymes that your cells have. And the RH marker is a protein. Which markers will you make? Well, that depends on your DNA instruction book, and so your genetics. So again, you can have all the markers or none of the markers, but whatever your blood type is, all your blood cells will have the same blood type. So your blood type is based on what markers you're born with. So if you have all the markers, you're called AB positive. If you only have the B marker, you're called B negative or BRH negative. If you're O, it means you lack the A marker and you lack the B marker. And so basically that's how you're named: AB negative, A positive, O negative, B positive, depending on which markers you make. So the markers on your blood determine your blood type. And so we have this A, B, O system, and then the positive and negative refer to whether you have the RH marker or not. So what about blood donation? Well, some people can donate blood to each other, and sometimes we have to be careful if there's a mismatch. So let's try to figure out whether people can donate and what are the rules with who can donate to who. Obviously, you can always get your same blood type, but we may want to be able to donate blood types that aren't exactly the same but are compatible. So again, in this example, let's make you A positive, which means you're born with the A marker and the RH marker on your red blood cells. And other cells in our body have their own markers as well, but we're going to focus on erythrocytes. So remember, your immune system, your immune cells, they can look at the markers on your cells, and we tend to ignore the markers that we're born with. So our immune cells will ignore the markers that we were born with and only pay attention to markers that we weren't born with. So what happens if you're exposed to a marker that you're not born with? Well, your immune cells, for example, let's say you have a bacteria with little B markers, and so your immune cells recognize those as non-self, not belonging. And so your immune cells can make defense proteins called antibodies, and those defense proteins will be specific for that marker that you were exposed to, in this case, the B marker. And so you'll have B defense antibodies. Now remember, your own cells don't make the B marker, and so those defense antibodies will only protect you against something that doesn't belong in your body. So now let's see how that would affect you with a blood transfusion. So let's say you're given donor red blood cells that are B positive. So B positive red blood cells have the B marker and the RH marker. Now you're born with the RH marker in this example, so your body would ignore that. But you've got those B defense antibodies which will recognize the B marker on those red blood cells and actually cause clumping and possible destruction of those red blood cells. So you can see in this case this would be a mismatch, giving the A positive person B positive blood. So one of the rules to remember is that your immune cells will tend to make antibodies, defense proteins, against the markers that you're not born with. Basically the markers that you don't have. So let's say you're O positive. If you're O positive, you make the RH marker. So now what kind of defense antibodies will your immune cells make? Well, in this case, since you're born without the A and you're born without the B, your immune cells will make A antibodies and B antibodies. Again, defense proteins that will seek those out whenever you're exposed to them. You would not make RH antibodies because you were born with the RH marker. Again, you don't want to attack yourself. You only want to attack foreign things like foreign cells or bacteria. So we don't want to give an O positive person any A or B blood. All right, let's look at some examples to see if you get the rules. So remember, the donor is the person giving the blood and the recipient is the person receiving the blood. So let's say you're B negative. So if you're B negative, it means your little red blood cells have the B markers, and that's it. So now your immune cells can make defense antibodies to the A markers since you weren't born with those. So you have A antibodies, A defense antibodies. And if exposed, you could also make RH antibodies as well. So let's donate some blood to this person. So we're going to donate AB negative blood, and let's see if that would react. So we put the AB negative blood in the body with the A markers and the B markers. Well, there's A defense antibodies which will attack and seek out those red blood cells, clumping them, lysing them, and destroying them. Could even form blood clots, which would be bad. So that was a mismatch. We wouldn't want to do that. Let's say you're A positive with the little A markers as well as the RH markers. and we donate O positive blood to you, which has the Rh markers. Well, in this case, if you notice, your body has those same little Rh markers on the A positive blood, so that's a match. And even if you're making B antibodies, there's nothing to react to, and so that is a match. You can actually put O positive, O negative, and the other blood types in that same person. What if you're B negative? So you just have the B markers. In this case, your defense cells will make antibodies against the A marker, so we don't want to give you any A blood. But let's donate some B positive blood and see what happens. So again, B positive has the B marker, but also has the Rh marker, which you weren't born with. So when you add that blood to your body, the B markers are not a problem. Your immune system would not react to that. However, you do not have the Rh markers, which we now have donated to you in the B positive blood. In this case, your immune cells will recognize that as foreign and will produce Rh antibodies. Again, defense antibodies against the Rh marker, which would then go after and cause clumping and lysis of those red blood cells. And your body would have those defense antibodies. So that was a mismatch. This becomes interesting if you have a mom who, for example, is the mom is A negative, and her little fetus, because of the genetics of the mom and dad, is A positive. In this case, now the red blood cells of the mom and the baby have a different set of markers. So the little fetal red blood cells normally don't get into the mom, but they can sometimes, and those little fetal red blood cells have the Rh marker. Because the mom's immune system has never seen those and they're foreign, the mom can actually make antibodies against the little fetal red blood cells. So in the case of a mom and a fetus, if the mom is Rh negative and the fetus is Rh positive, it can actually cause this hemolytic disease, which can cause destruction of red blood cells in the fetus, causing loss of the pregnancy or sometimes an injured baby. Okay, see you guys in class.